Can somebody please confirm you can hear me?
I can hear you.
Thank you. Before we begin, are there any questions? All right, not hearing anything, so let me begin. Let me state that there was a problem in the last quiz for about some of you. There was a question that had the wrong answer in the quiz, so it was automatically graded incorrectly. I've now changed it, and I gave people, even if you got the wrong answer, you got that question correct. So at least that's the way it should be. I did tell it to accept both the new answer and the old answer, so you should have gotten that question correct. I think it was question 5, which actually I think was of the exam, but actually the number was 4, something like that. And the question numbers are a little bit off because I added question 0 and it doesn't count with a 0. All right, any questions about anything? Oops, did I drop that? No, there it is. Are you seeing my screen here?
I can see your screen, yes.
All right, thank you. Um, Today is February 12th, and we should be doing Lab 8, identification of gram-positive organisms. You should also be working on your unknown work, and I should too. I meant to look that up yesterday, and I never did. So let me look on the unknown notes pages and see if anyone's got anything. I'll promise to do it after the lab today. Um, let's get to Lab 8, identification of the gram-positive organisms. Oops. Oh, by the way, my computer is going to be very slow because we're still, I can't even show that. I don't need that. We're still working on the lecture. Oh, there it is here. It's 90% done, the lecture page. Why is it slowly stopping at 90? I don't know. So just bear with me until my computer starts speeding up. Lab 8, identification of gram-positive organisms. We're going to be talking about gram-positive cocci, and that's in the wrong place. That's better. Still in the wrong place. Ah, I'm not sure how to get this. Anyways, we were talking about gram-positive cocci, staphylococcus, and streptococcus. Get that back. I don't know what to do here. Oh, good. The lecture at least is finished. I'll come back to where I was. Anyways, the streptococcus should be over here. Didn't seem to go. The enterococcus should be over there as well. Hopefully you guys can make sense of this. Listeria is a rod, so it should be over here. Oh, why did that move? Right there. Rhinobacterium is a rod, Bacillus and Clostridium are all gram-positive bacilli. Kind of a mess here, but at least now it's kind of correct. Hopefully yours in the lab module will not be as messy as this one. There are some video clips as a supplemental material today. We're going to have some case studies and a gram-positive dichotomous key. The last two are provided as separate files in Canvas, so let me show you that now. Oh, I need to close that down. Well, that's funny. It just closed down. So let's scroll down today's lab. Actually, it was added, but I had to go up to look at the something for a student. Lab module 8. Is that the one we're on? Yep. All right. So there's the lab module. There's the worksheet for lab 8. And then you have some case studies. 1, 2, 3, 4, 5, 6, 7, 8. And one dichotomous key. I thought about putting all these together in one page, and I'm glad I did not, because the person who sent them to me later made corrections on case study 4 and 5, and that's why they have a little different name on them. And so I'm glad I have them as left independently, and I kept them independently because the person who sent this to me is now he's the chairman of the department. So if he wants to make one change, just put up the new case study. But to do the case studies, you have to click on each link, case study 1 through case study 8. And then to use the dichotomous key, you have to click on this link. These are not in the lab module or in the worksheet lab 7. Excuse me, lab 8 or the worksheet lab 8. All right. The learning objectives of this lab: 1, understand the principles and how they interpret the results for the CAMP test, the Bialasculin auger slant test, the SF broth test, and the optogen sensitivity test. And then 2, be able to use a dichotomous key to solve case studies involving gram-positive bacterial infections. Let me mention that this dichotomous key can only be used for this lab. Do not use it for your unknown project. Do not use it for any other lab. This dichotomous key is specifically designed for this lab and this lab only. When specimens are received in a microbiological lab for routine bacterial cultures, meaning a culture is sent to a lab to find out whether the patient is sick, the cultures are inoculated on a culture media that will facilitate the growth of common pathogens and placed in a physical environment that will support their growth. Microbiologists can use colony morphologies and the gram stain to provide important information in the primary classification of a bacterial isolate. However, further biochemical testing is required in order to isolate the species which is making the patient sick. In this lab, we're going to talk about the CAMP test, the Bialasculin hydrolysis test, the SF broth test, and the optogen sensitivity test. Any question about any of that? All right, the CAMP test is named after the 3 microbiologists who invented the test. So it's an acronym for Dr. Christie, Dr. Atkins, and Dr. Munch Peterson. C for CAMP, A for AMP or CAMP, MP for MP of CAMP. They first described the amplified hemolytic effect that some beta hemolytic organisms have when they're grown physically near Staphylococcus aureus. What you do to do the CAMP test is you streak a single streak of beta hemolytic Staphylococcus aureus down the middle of a blood agar plate. So right here, right there is a streak of Staphylococcus aureus. And as you can see, the darkening here, almost orange there, but darker here, is the streak of Staphylococcus aureus. And around it, you can see a clear region, and that's from beta hemolysis of Staphylococcus aureus. You then, perpendicular to that Staphylococcus aureus streak, shrink that down a little bit, a streak an unknown or test organism, which is also beta hemolytic. And what you're looking for is to determine whether in the CAMP test you have enhanced hemolysis near Staphylococcus aureus. We'll let it way up. So right here we have Streptococcus pyogenes, and there you can see the dark streak in the center here. That's our growth of Streptococcus pyogenes. The lighter region around it is from beta hemolysis of Streptococcus pyogenes. You'll note that the hemolysis here and the hemolysis here near the Staphylococcus aureus are the same. So Streptococcus pyogenes has no enhanced hemolysis of the growth near Staphylococcus aureus. Any question about any of that? And that would be negative in the CAMP test. No enhanced hemolysis of the growth near Staphylococcus aureus. Let's streak over here, the Streptococcus galactiae, and there you see the streak of Staphylococcus aureus, the hemolysis of Staphylococcus aureus, and there in this dark line, which actually is two lines right there, we see Strep galactiae growing, and there you can see the clear region around it, the beta hemolysis around Streptococcus galactiae. And you can see here greatly enhanced hemolysis in this region here of Strep galactiae near growth near Staphylococcus aureus. And then further away, we have no enhanced hemolysis. Any question about any of that? Now note that the CAMP test is always working with an organism which does perform beta hemolysis. What we're looking for is enhanced beta hemolysis near Staphylococcus aureus growth. And so this would be CAMP positive, by the way. You also have some enhancement, slightly positive CAMP test, where here we have Listeria streaked here. You can barely see the bacteria, the dark reddish streak there and there. And then you can see around it very slight beta hemolysis. Oops. Well, what happened to my screen now? Enhanced hemolysis right there, that's beta hemolysis. And then right here, slightly enhanced hemolysis right there in the Listeria growth near Staphylococcus aureus. So this is a slight enhancement of beta hemolysis. So Listeria, slightly positive, Strep galactiae, positive, and then Strep pyogenes, negative in the CAMP test. Any question about any of that?
So when we say it's positive, we would just say enhanced beta hemolysis or the arrowhead. But if it was negative, it would just have nothing.
Correct. Nothing is Streptococcus pyogenes. It has no enhanced hemolysis.
Okay.
So that's negative in the CAMP test, positive in the CAMP test, and this one's slightly positive in the CAMP test. Slightly enhanced hemolysis. And I guess you have to look at all three of them to see slightly positive. And you're right, it normally forms an arrowhead in a positive camp test. Even the slightly positive camp test is a arrowhead. And I don't know why it sure is the arrowhead, but it's always an arrowhead. It's probably because the Staphylococcus aureus is using resources near Staphylococcus aureus, and so there's less enhancement as you get closer to Staphylococcus aureus. And then right here, you're getting diffusion of the ingredient from Staphylococcus aureus coming out. And beyond that point, we don't have any diffusion, so there's no enhanced hemolysis after this part of the arrowhead. But right there, we have maximum enhancement of hemolysis because of diffusion from Staphylococcus aureus, making maximum hemolysis. And as you go in, there's competition with Staphylococcus aureus, and so there's less enhancement. That's my guess for why there's an arrowhead. Anyways, any question about the CAMP test? If not, let's move on to the bile-esculin hydrolysis test. Bile-esculin is an agar slant used to differentiate between Enterococcus and Streptococcus families. If the organism can hydrolyze esculin, it will produce esculatin, and esculatin has a dark brown color, and esculin is broken down into glucose and esculatin. And if the dark brown precipitate occurs in the tube, it is positive in the esculin hydrolysis test. And if the esculin does not break down, it'll be its normal color, kind of a light brown color. So that's esculin negative or esculin hydrolysis negative. Streptococcus species are bile-esculin negative. Enterococcus species are esculin hydrolysis positive, meaning they'll be dark brown. Any question about any of that? All right. The SF broth test is named after a species that used to be called Streptococcus faecalis, but when they sequenced the species, they realized it's not in the Streptococcus family, that is actually in the Enterococcus family. Is that clear? So what used to be called Streptococcus faecalis is now called Enterococcus faecalis. However, the test is still called Streptococcus faecalis, and I'm just going to call it the SF broth test for short, so that way you don't even need to know. The old name of the species used to be Streptococcus faecalis. In the SF broth test, you inoculate the media, which is purple, and if the organism can grow in the tube, it'll make acid products, changing the pH of the indicator from purple to yellow. So in the SF broth test, this is positive yellow color, and this is negative color, meaning it remains purple. All members of the family Streptococcus, and we're talking about the species now named Streptococcus, are negative in the SF broth test. And a species which is positive in the SF broth test is Enterococcus faecalis, which, remember, that used to be called Streptococcus faecalis. You don't need to know that, but the point is Enterococcus faecalis is positive in the SF broth test. So this test can separate out Enterococcus faecalis from members of the family Streptococcus. Any question about any of that? All right. The optogen sensitivity test is used to differentiate between Streptococcus pneumoniae and other alpha hemolytic streptococci. So we have a bacteria here, which you can actually see here. Let me blow that up so you can see it better. It's hard to see because it's a little dark. Let me blow that up to 300 times. There we go. There is the species of bacteria colony there, and it has a darkening around the species. Or there, right there is the colony, and it's got a dark ring around it. The dark ring is from alpha hemolysis of the blood agar. The blood here would be this light red, and there's alpha hemolysis. This whole streak is alpha hemolysis. And we're looking specifically at these just because we are. It's easier to see here. And you then take the bacteria, which has alpha hemolysis, and then streak it on the blood agar plate here. Shrink that down now. And then stick the optogen disc, which is the P standing for optogen disc, on the blood agar, and then incubate the plate. If there is a region showing that the bacteria are sensitive to the antibiotic optogen, meaning the zone of inhibition is 14 millimeters or greater, then the bacteria are said to be optogen sensitive, and a species which is optogen sensitive is Streptococcus pneumoniae. If, on the other hand, the bacteria is alpha hemolytic and is a member of the Streptococcus family, then it will be optogen resistant. And by resistant, you either have no zone of hemolysis or a zone of hemolysis less than 14 millimeters. And that will be a member of Streptococcus family because we identified it to a member of the family that is alpha hemolytic because it's showing alpha hemolysis here, but it is not Streptococcus pneumoniae. It's some other member of the Streptococcus family, which is alpha hemolytic, because it is not optogen sensitive. Any question about any of that?
Okay, so it can only be an option like positive when it comes out that there's a zone of inhibition or if it's less than the 14 millimeters, but only in that scenario. But if it's less than 14 millimeters, like right above when it says there's other types of alpha hemolytic
There are other
demonstrates the resistance.
species of Streptococcus which exhibit alpha hemolysis, other than Streptococcus pneumoniae. I can't remember one off the top of my head, but you can actually look at the table of the unknown, and it's got at least two species of Streptococcus which are alpha hemolytic. Okay, and there's others which aren't listed in the table, but there's at least two that are alpha hemolytic. And
Okay.
if it's optogen resistant, meaning it has no zone of inhibition as this, or the zone of inhibition is less than 14 millimeters, it is a member of Streptococcus, which is alpha hemolytic, but it is not Streptococcus pneumoniae, because Streptococcus pneumoniae is a member of the Streptococcus family, which shows alpha hemolysis, but it is sensitive to optogen.
So can this test only be used to determine if it's pneumonia?
Yes, you got to remember that we've narrowed it down to the family of
Okay.
Streptococcus and then narrowed it down to only the alpha hemolytic Streptococcus. So there's really only a few that
Okay.
it can be. And I don't remember how many, but there's a few species of Streptococcus which are alpha hemolytic. It's not a whole lot. It's certainly less than a handful. It's either 2 or 3 or something like that. Okay?
Okay, thank you.
All right. So this main test is really to determine that it's Streptococcus pneumonia. Yes, that's the purpose of this test. All right. Microbiologists use the dichotomous key when they're performing tests to decide which organism they have, meaning they use dichotomous logic to go through the test logically. First, we narrowed it down to family Streptococcus, and that's not shown how we did that. And then they narrowed it down to alpha hemolysis. And then they narrowed it down to, let's talk about Streptococcus pneumoniae, sensitive to optogen, or resistant, in which case it's not Streptococcus pneumoniae, it's one of the other alpha hemolytic members of Streptococcus. That's using dichotomous logic. And I don't know how they did obtain how we know it's Streptococcus because the tests were not shown here, but we have that narrowed down that the species is a member of Streptococcus family. And that's using dichotomous logic. Other options are possible. You can take a look at that if there's a link. But dichotomous logic, you start with all species possible, and then you can do the gram stain, separating all species into two, the gram positive and the gram negative. If it's gram positive, we can then scratch out the gram negative and then proceed further with the gram positive. The next test we did was the Cadley's test. And here we got Cadley's positive cocci. Oh, wait a minute. Sorry. The next test we did was what's the shape of the bacteria. And we got gram-positive cocci and gram-positive bacillus. The bacteria is gram-positive cocci, so we're going to continue with this one. And then we cross out the gram-positive bacillus. The next test we'd run is the Cadley's test. So the gram-positive cocci can be Cadley's positive or Cadley's negative. If it's Cadley's negative, it's the member of the Streptococcus family or the member of the Enterococcus family. And you could do the SF broth test, and then you get this member of the Streptococcus family. And that's probably how the people obtained that this bacteria was Streptococcus, by the way. So they went through dichotomous logic and they discovered this species is a member of Streptococcus, and then they streaked it on blood agar and got that it was alpha hemolytic Streptococcus. Anyways, here we're going to now scratch this out. There's not Cadley's negative, it's Cadley's positive, and that means it's of the family Staphylococcus. You can then do this coagulase test. If it's coagulase positive, there's only one member of the Staphylococcus species, which is coagulase positive, and that's Staphylococcus aureus. So I've now identified the species because I'm going to say it's coagulase positive. If it's coagulase negative, it's another member of Staphylococcus species other than Staphylococcus aureus, because all the other members of the family of Staphylococcus are coagulase negative. Okay, any question about any of that? Anyways, that's how you use dichotomous logic, where you start with all bacteria. You then do a test separating the possibilities into about two possible. Then you cross one out, then follow this one. You do another test, separate into two possibilities, the bacillus or the cocci. Cross one out, follow the cocci into catalase positive, catalase negative. Any question about any of that? Someplace we have a dichotomous logic. Maybe it's this link here or that link there. There's someplace in this lab if you want to go through using dichotomous logic, you can. If you understand dichotomous logic perfectly, you do not need to use that link and go through it. All right, you're not going to be physically performing any of these activities because everything we do will be done online. However, you need to be able to be responsible for knowing how to perform and interpret the following tests. So you view the video clip on SF broth test and the bile-esculin test. And this is just one link. We'll talk about both. And then view the link for viewing a video, short video on discussing the CAMP test. And then let's go ahead and go to the worksheet now. Review how to interpret the following tests by completing the chart. For some of them, you can get the information from the lab module in this lab. For others, you will need to refer to a previous lab to get the information for filling in the table here. Like for example, the gram stain, you can get that from a previous lab. I've already given you the results. The gram positive result, you say it's purple. The gram negative result for the gram stain is pink. So give me the result for the gram stain. Give me the result for the Schaefer-Fulton stain for endospores. Give me the positive result. Give me the negative result. Give me the negative stain for the capsule. What you see, give me the positive result. And if you need to tell me in a paragraph, just expand this box, write in your answer. With the endospore, you could say something as simple as a clear zone around the species that is showing the capsule. So you just say clear zone around the cell. Give me the positive result for the catalase test, hydrogen peroxide, putting the bacteria in hydrogen peroxide. And all you need to say is bubbling or not bubbling, or you can say catalase positive, catalase negative, either one. For the coagulase test, give me the results of the coagulase test. You could say forming a clump or liquid stays liquid. And then you could also do forming clump splotches on the slide. And then the other would be a smooth, what do you call that? A bacteria suspension on the slide. You can just say clear bacterial smear or smooth bacterial smear. Give me the positive result for the oxidase test. Give me the negative result. Give me the positive result for the CAMP test. You kind of need to describe it. Enhanced hemolysis near the Staphylococcus aureus. You could just say enhanced hemolysis or not enhanced hemolysis. Bile-esculin, this is in this lab module. Give me the positive result. Give me the negative result. For the SF broth test, give me the positive result. Give me the negative result. For the optogen sensitivity test, give me the positive result. Give me the negative result. For the basis tracing sensitivity test, give me the positive result. Give me the negative result. Optogen is discussed in this lab. Bacitracin is not discussed in this lab module, but you need to know it because we talk about the bacitracin sensitivity test in one of the questions in the worksheet. Here's the link for viewing a video on how to develop and use a dichotomous key. You do not need to construct a key for this class. However, the video will help you understand the general idea and concept behind making a dichotomous key, which you will need to make for your unknown bacteria. So your unknown project. So if you're a bit confused, go through this link, But if you're very comfortable with the dichotomous key, you don't need to click on that link and go through it. In 3B, we have the bacteria that we're going to be considering in the dichotomous keys, which you can find in the link, which is shown here. Can you guys see this? There's the link to the
Yes.
dichotomous key. And then work on the case studies. Case study 1, put your answer here where you list your species name. So you need to give a species name right here. And then when you're all done with all 8 case studies, submit this document to the normal place in Canvas to submit the worksheet. You have to submit the worksheet because it only has the questions. But for case study 1, you come back to here, click on case study 1, read it. Actually, it'd be easier for me just to open it here. Oops, wrong one. Here we go. Case study 1. A 13-year-old teenager, shown here, attended a week-long summer scout camp, also shown here. Upon returning home, his mother notices a pustule beginning to develop in a spider bite in his arm. The area is warm to the touch and is becoming extremely painful. You can see it red and swollen right there. When her son starts running a fever, she takes him into a walk-in medical clinic for evaluation. The official orders a gram stain of the exudate and a routine culture. Here are the findings. Let me scroll down just a little bit. There you can see the gram stain. There's the bacteria there. The big pink cells are actually his cells. And so you have to blow it up to take a look at what we're looking at. You can actually see the shape of the cells and kind of the growth arrangement as well. And then they grew the bacteria on blood agar plates, and you can notice something about the blood agar plates right there. They then took the bacteria and added it to hydrogen peroxide. I'll tell you that this is the catalase test, and there's the result there. So get your results from the catalase test. Write it down, I guess. And then they do take the bacteria, put it on the slide here, add rabbit plasma on top of the bacteria. After 15 seconds of rocking, there we have the result there. Write down the result of the test here. You can look up the test if you don't know what the test it is. And then we do test 3. We're growing the bacteria on a McConkey's. Nope, it's MSA. Sorry, MSA plate. And this bacteria is negative. That bacteria is positive. Both bacteria can grow on MSA, so they grow in 7% sodium chloride. But this one, you're seeing something happen which changed the color of the plate from the negative. So write the result here for test 3. And like I said, that test is the MSA test. For test 4, we have the Nova bias and sensitivity test. We took the bacteria here and then streaked it out on the plate so we have bacteria growing as a lawn. And before we incubated it, we put down Nova bias and antibiotic disc there. And there is the result. And take your results and compare it to the chart of the dichotomous key shown here. And gram stain. Anyone note, was it gram positive or gram negative?
Positive.
Positive cost.
It's gram positive, so that's correct. And actually, this is gram positive here, and scroll down a page, there's gram negative. So we'll cross this one out because it's not gram negative. And then is it cocci or bacilli?
Cockeye.
It's cocci. And so that splits it there. And then you continue going down this way until you have identified the species. And that's how you use the dichotomous key. Any question about case study one?
For the third result, the photo from the rabbit test, that one it was kind of hard to tell what was happening in the picture because I know that the results should either be clotting or clumping or not, and I couldn't really tell from the photo if that was a clump or liquid form.
This is not a tube test. This is the slide test. And where do we have that shown? Let me look the lab module.
The test two that
We
you were
didn't
just
test
that
this.
one,
We
yeah.
didn't do this. We didn't do this test today. So you got to look at the previous lab for the coagulase test. Anyone know what lab module that's in? Coagulase test?
Was that in the urine and throat
Yeah,
module?
it was week 7 or
Week
lab 7.
7? That's where the catalase test was up above. Up, here we go. Scroll up just a little bit. So this is the slide test, and that's the tube test. We are doing the slide test. And so did it look negative or did it look positive? Okay.
Negative. Gotcha.
Yeah, I didn't want you to mention the answer, but that's all right. Um, all right, then when you have the species identified, write it here, and then move on to case study two. Let me just mention, I've told you test two, it doesn't say what test it is. It's the basic trace and sensitivity test. So let me just go to case study two so I can point that out. Oh, I'm no longer on that page anymore. Case study two. All right, a 27-year-old female receiving prenatal care. She has no symptoms or anything like that, but the doctor just did routine culturing and he discovered something here that kind of alarmed him. And he did a gram stain on it. He did, once again, the coagulase. No, this is hydrogen peroxide test. So this is a catalase test for test one. Test two, it doesn't state it here, but they then took the bacteria grown here, put it on this media, and put an antibiotic disc on. And I'm telling you that that test is the basotrace sensitivity test. And you can see no zone of inhibition around basotracin, so you should be able to get the results to that. And then they did the camp test here. You can see the camp test right there. And then what species was isolated? Go ahead once through the dichotomous key. I think it's gram
Wait,
positive.
so
Hmm,
have we gone over the basic tracing sensitivity test in a previous lab module?
I think so.
You- Uh, does anyone recall what lab module that might be? Because I don't remember that test.
I think it was
Let's
either
see. Where did we
6
talk
or
about?
7.
Where did we talk about antibiotic resistance? I think that's where it was.
It was
Oh,
the
wait.
urine one.
Oh, okay.
The very bottom, I believe.
We
of it.
did a whole lab on antibiotic resistance, and you're saying it's in
Right,
that
right.
lab module?
Bacitracin, I believe, was one of the bacteria studied in that lab. Is that correct? Does anyone? No.
I believe so, yes.
Yeah. Um, look at that to decide whether it's bacitracin sensitive or bacitracin resistant. Get your answer, and then use that to help you in the dichotomous key. Where do I have that right here? Oh, let's go there again.
Wait, so is bacitracin an antibiotic, or is it a bacteria?
It is an antibiotic, bacitracin,
Okay.
and it shows up someplace here. Right here. And so you can use the bacitracin test and the CAMP test to separate out Streptococcus pyogenes from Streptococcus galactiae. Yep, that's the only place it's used.
And you said the last page for the little key was actually gram negative, but we can still use that, or we just don't use that entire last page.
No, the last page is gram-positive bacilli.
I'm sorry, I thought you had said this is what was all gram negative. My apologies.
No, everything we do today is gram-positive, but there's gram-positive cocci, which is the first page, and then the second page, which is gram-positive bacilli. Let me see if I can scroll down there. No, I can't scroll down there any further. Oh, here we go, right there. Okay, so the second page is gram-positive bacilli. There are some species of the in the case studies which are gram-positive bacilli. So you will need the second page. Okay? All right. If there's any further questions, I will be here until 8 o'clock or the last student logs out to help you with the lab. So if you have a question, make sure you ask before the last student logs out. Any questions?
Is it okay if I maybe email you a question? I have to give my daughter a bath and cook her dinner, right?
All right. Somebody asked, how do you answer the question in 3B? For 3B, there is no question, so you don't need to do anything for 3B. 3B, all it is doing is listing the species and telling you that these species are in the dichotomous key.